So I gave this presentation the title, The Exploratory Potential of Volume Electron Microscopy. Um, and simply because I was quite excited by some of our recent results and also by, screening some of these large open access volume EM data sets. But I want to start to giving you a bit of an overview on what we are interested in. And as Johanna already mentioned, I'm interested in insulin secretion. And most of you will probably know that insulin is made and stored by the beta cells. And these are the endocrine cells of the islets of Langerhans. And you see one of these islets here on the right-hand side. And these are microorgans that are embedded in the pancreas. And they are really little islands within the sea of exocrine tissue. The beta cells are not the only cells in the islets. There are also the alpha cells that secrete glucagon. Delta cells that secrete somatostatin, so there's a balance between these different hormones. And islets are vascularized and also in connection with the nervous system. When we now focus on one of these beta cells, and this has been the focus of my work for the past more than 10 years, you see here an EM image of one of these beta cells, and you see the nucleus in the center, and all these smaller vesicles, these are insulin granules. So these dark spots are insulin cores. I just highlighted some of the granules here in orange. So these cells are really filled with insulin. And now when blood glucose levels are increasing, the cells take up the glucose. There is an increase in the ATP-ADP ratio, closure of the potassium channels, depolarization of the membrane, calcium influx, and this then triggers the release of insulin by fusion of some of these granules with the plasma membrane. Also, the cytoskeleton plays a role in these processes, and we are in general interested in how insulin secretion is regulated, also on a structural level in the beta cells. So I will talk about two main projects, and then I will give an outlook on what we are planning in the future. And all of these projects are heavily connected to volume EM. But one of the first questions we already had some years ago was, how do beta cell microtubule networks actually look like, and how do they interact with insulin? Because there was a bit of the discussion if microtubules are actually helping in transporting insulin to the membrane, or can they also act as a barrier within the cell? Then we move on to a broader tissue context and looking into beta cell cilia and how they interact with the islet and non-islet tissues. And then we move even into a more broader context, asking the questions, can we map all of the connections within one of these islets of Langerhans? And we start now with insulin transport. And what you can see here is actually a live cell structured illumination movie with the microtubules in green and insulin granules in magenta. And you see that one of these insulin granules here has been traveling on microtubules. So we know that granules use these microtubules to travel through the cell. But as I mentioned before, there was a kind of discussion on really the role of microtubules. Are they positively associated then with insulin secretion, or can they also form a barrier, especially when the cell is not stimulated? And at the time, we did not have a full view on microtubule networks of beta cells. And this is why we actually turned to volume EM. And this is just a quick reminder that volume EM does not mean only one technique. It is like an umbrella term for various different techniques of TEM or SEM methods, more or less automated. And some years ago, we started this collaboration with Harald Hesse's lab at Geneva, and Shan Xu and Song Pang, and Shan and Song are actually now at Yale, where they have set up the enhanced FIP that Shan developed originally at Geneva. And we have been using the enhanced FIP, meaning allowing imaging of large volumes at high resolution for looking into beta cells and also resolving microtubules in complete cell volumes. And I just want to show you one of the raw movies here. So this is from isolated islets. So these are digested out of the pancreas. So we lose the blood vessels and the innervation, but we still have the endocrine cells, so the beta cells and also the other cells of the islets. And this has been fixed by high pressure freezing and imaged at 4 nanometer voxels. So when we now go through these volumes, we are now at the nucleus. We see all these spherical objects are insulin granules with the insulin core. We see mitochondria and also the endomembrane system really well. And in the still image, you see that we could also resolve here the microtubules. And this was now published in 2021, and the project started like 3 years earlier. And we were then thinking about how to actually analyze those data. So we had two large FIB-SEM data sets. And at the time, I used a variety of different segmentation tools. This is all now published also in a protocol. And you know that especially segmentation tools have heavily evolved in the recent years. So probably one does not need to do it like this anymore. But this also included some manual segmentation of the microtubules, semi-automatic segmentations in microscopy image browser. And you might know that actually MIB is capable of doing many more advanced things, including deep learning, and then random forest segmentation of the mitochondria in Elastic, and then also some deep learning for the vesicles. And in this talk, I don't want to focus on segmentation. And you know, there was this Christmas talk by the colleagues in Czech Republic that also included a lot of advice on how to do segmentation. This is now online on YouTube also, so I highly recommend you to look into this. I will just use like 1 or 2 slides to talk about Album, because this is a decentralized tool that allows you to perform different tasks, including segmentation, analysis, and also 3D visualization within this one platform. And we use this for the analysis and also for plotting of the data. And also we have a solution for getting our segmentation masks into a nice Blender file. And this was originally developed by Kyle Harrington, Deborah Schmidt, and Jan-Philipp Albrecht in Berlin. And it's always a bit tricky to explain, but it's really a centralized platform that can have access to open source software tools, and these can be segmentation tools or analysis tools or visualization tools. And then you can create solutions for your task. And I just will run one of these videos here. So this is the GUI. And for example, we have now one solution for running a spatial analysis. And I won't show you the whole video, but we will just have a look how this works. So before we actually can run our solution for the first time, we will install all the dependencies, and this happens automatically. So you don't have to search manually for some things that were missing to run the code, for example. So this is quite nice. And we have here our project that we have already created doing some of the segmentation tasks. We are now, for example, defining a threshold for defining organelles as connected in our case. And once this analysis has been running, this will open a big data viewer window where we can look at the segmentation masks. And we just will wait now some seconds for this to open. And this allows us then to look into the connections of different organelles, for example. And we can then also create solutions for the 3D visualization. And also like plot these analysis results. So this is also part of the protocol that we have published. So if you are interested, you can take a look into this. And also Album is not really targeted only to our problem, but you can create and run solutions for a lot of different image analysis tasks. And now this big data viewer file, we can now look into different parts of our segmentation masks. And we can also look, for example, into insulin granules in this case that are connected to the membrane or that are close to the nucleus. And we can look into these subsets. And then in the next step, we can also create plots with the distances, for example, between these different organelles. So I'm jumping now to the next video that is somehow summarizing the results from this project. So the goal was really to look into the microtubule interaction with granules and also basically to get an idea how the microtubule networks in these cells look like. And this is now a rendering in Blender of one of these beta cells where you see that they are really filled with organelles. So there's basically no free space. And you get an overview on the organelles we have actually segmented in this project. So we have the mitochondria here. We saw the insulin granules previously, the Golgi, and then these weird kind of microtubule networks. And just to summarize, out of these two large FibSEM data, we were able to reconstruct 4 cells under high glucose conditions. So this would be stimulation with glucose that induces insulin secretion, and then 3 cells under low glucose, which would not induce insulin secretion. And just looking into some of the basic data that we got here, of course, now after segmentation, we can do simple counting, for example, of the insulin granules, and SG stands for secretory granules. And you see that we have quite a big variability in the granule number, right? We have some cells that have 4,000 granules. We have a cell that has almost 14,000 of these granules. And then looking into the microtubules, when we count those, we saw that in the low glucose samples, we have fewer microtubules compared to the high glucose, which was kind of interesting because it seems that there's something going on after the stimulation with glucose. We can get also organelle volume fractions out of our data. And this also shows again this kind of heterogeneity, but this is not happening for all of the organelles. We see heterogeneous volume fractions for the insulin granules and also for the mitochondria. However, the volume of the nucleus and also of the Golgi is pretty stable in our case. Then when we look into more detail, of course, we do not only want to count organelles, we also want to really measure them, right? So when we look into the length of the microtubules, we saw a difference between low and high glucose. So in the lower glucose samples, where the microtubules are fewer, they are much longer compared to the high glucose. So there might be really some remodeling here happening after this metabolic stimulus. But we also wanted to get an idea on how these microtubule networks are actually organized. And from these textbook images of, for example, HeLa cells, we have this perception of a microtubule network as like a radial network, like spokes in a wheel, right? But a lot of cells in our body have specialized networks, for example, in neurons. And these are actually non-centrosomal. And we saw this also in beta cells, that actually most of the microtubules in low as well as high glucose conditions are not connected to the centrioles anymore. Then there was the idea that the Golgi apparatus can generate microtubules, and it could also in our case, because still some of the microtubules are connected to the Golgi, but also here not the majority of them is. So really 90% of the microtubules in beta cells are not connected to the Golgi or to centrioles. So really this is a distinct non-centrosomal network. So after looking into this architecture in general, we were interested in looking into the behavior of insulin granules in the beta cell, and then afterwards to look into the connectivity between microtubules and insulin granules. And just looking into insulin granules in general, this is what you can see here. So you see a cell here with the nucleus, the insulin granules, the Golgi, and the plasma membrane. And with our data and also within the album tools, we can now plot the distance of our organelles, in our case, the granules, to the plasma membrane. And this is what you can see here on the right-hand side on the top. So we have here the distance to the plasma membrane in microns, and we see here the percentage of insulin granules that is at this certain distance to the membrane. And this black line here indicates actually a random distribution. So this would happen if our granules would be randomly distributed within the cellular volume. And what was interesting in this case is that we found this peak here in low and in high glucose conditions close to the membrane. So granules are enriched at the plasma membrane, and this is not really connected to glucose stimulation. Whereas when we look into the granule distance to the nucleus, and this is what you can see here on the lower left, this distribution follows a random distribution, And then when we look to the lower right, we see the distribution to the Golgi apparatus, and we see a little peak for the high glucose. And this might just mean that the granules, the cells are producing more insulin because they are also stimulated, because the stimulation does not only lead to insulin secretion, but also to the production of new insulin. So this was quite interesting. And then we looked into microtubule insulin granule interaction. And there we also saw an enrichment of microtubules and granules close to each other, which is highlighted here by this peak, basically at 0, right? And this is also not explained by random distribution. So in both conditions, the insulin is close to the microtubules. We also then defined a threshold for looking into connectivity, and we call it association because we cannot really speak about biological connectivity. But we found also that the majority of insulin granules in the periphery of the cells is really very close to microtubules. And this is what you can see here in this video. And I just want to quickly sum up the results here. So we see this non-centrosomal network that is also not connected to the Golgi. We see this interesting alteration in microtubule length, which means that metabolism influences really the structure of the cells. When we see this enrichment of granules close to microtubules, also in the periphery of the cells. So the conclusion here was that microtubules most likely maintain an enrichment of granules near the plasma membrane. So in these data sets, we also saw something that we have been actually interested in before. So most of our cell types in the body have a primary cilium. And this is usually non-motile, but it's an important signaling hub for receiving signaling from the outside, from the neighboring cells, or even from further away. The beta cells also have one, and this is what you can see here in one of these 3D renderings, that we have in purple here, the centrioles and the axoneme, which is the microtubule structure of one of these primary cilia. And we know that beta cells have the cilium, and it's important for paracrine signaling within these islets. So the beta cells can receive signals from the neighboring beta cells or alpha cells. And we also know from previous work from other groups that the dysfunction of cilia impairs insulin secretion and leads to a diabetic phenotype in mice. And there have been, the cilium is really a signaling hub, and it is isolated from the rest of the cell and also from the plasma membrane, and it's usually enriched in GPCRs, and some of these have been also identified already in beta cells. However, what we were missing was a complete 3D structure and also a comprehensive mapping of the interactivity of beta cell cilia with the surrounding tissue. And now we had already in our FIB-SEM data sets some cilia that we could detect, and this is what you can see here in panel A. So this is mouse FIB-SEM of isolated islets at 4 nanometers, and you see in the cilium is highlighted in orange. And you can see here also that 4 nanometers is probably not the optimal resolution for this, but you can discriminate the doublets from the microtubule singlets. And then we also wanted to get an idea on the structure of cilia in human beta cells. And this is now patient material here in panel B. And there we actually turned to serial section electron tomography, which meant that we then have to align these sections together after taking the tomograms, but we are more flexible with our resolution. So we could go down here to 1.3 nanometers, and you can appreciate here that with this higher resolution, it's much easier to discriminate the doublets from the singlets. But both modalities actually allowed for the reconstruction of cilia in these beta cells of mice and human. And I just want to run now one of the videos, and we start here actually in the FIB-SEM data at the tip of the cilium. So you see now these microtubule singlets. And the closer we get to the base of the cilium, we see that there are microtubule doublets that have been transitioning towards the center. And we can then use these data to do a segmentation. And in this case, this was actually done manually because we did not have many cases at this high resolution where we could actually do a segmentation. And this was actually done pretty quickly by manual segmentation. And just to show you quickly some of the data, we see this kind of disorganized axoneme that has been described also for primary cilia of other cell types. There are not so many full 3D reconstructions yet. And as I said, in the beta cells, we didn't really have this. And we see this termination of the B tubules here. This is highlighted by these arrowheads. And what we also can see in panel B, that some of these outer doublets are transitioning towards the center of the cilium. And we actually had this discussion from 2D sections that there might be a central pair that would be important for an active movement of these cilia. But these are actually outer doublets that make this transit towards the center of the cilium. And you see in C and D that we could also plot these data nicely, and you can see actually highlighted here this transition of some of these doublets towards the center of the cilium. Within this islets context then, so these, especially in the Fib data, right, we had the neighboring beta cells, we didn't have blood vessels, we didn't have nervous system, but we could look into the interaction of cilia with the neighboring cells. And this is important for their signaling capacities. And we started looking into our older Fib data, into these interactions, and this is what you can see here, that actually cilia are really squeezing in between these islet cells, and they have very long ciliary pockets in some cases. This is what you see here, but also once the cilium leaves its cell, it's immediately in contact with neighboring cells, and this is what you can see in this video here. So we see one cilium, we see this long pocket when we remove the inner parts of the cell. But also the outside of the cilium is then immediately in contact with neighboring cells, which gives them the opportunity to receive signals from these neighboring cells. So this is really then squeezed in between these narrow spaces here. We also could see some even more intense interactions, especially in the human samples, and we cannot really explain this yet, but we are looking into the signaling pathways that might be happening here. And what you can see here is in purple, one of these beta cells here that is really pushing the membrane of the neighboring alpha cell in orange. And it's making a little tunnel in the membrane. So it's quite an intense interaction. And we see this in several data sets now, and we don't know what it means, but there is a lot of options also here for signaling. And so now we have been looking into a bit of this isolated view on beta cells and also in isolated islets. And I'm now here back at the single beta cell. But when we look into this context now, we see that the beta cell is not alone, right? And in order for us to better understand beta cell function and insulin secretion, also hopefully dysfunction in diabetes, we need to look into a broader tissue context. And what you can see here is data from a large FIB-SEM data set that has not been generated by us, but by our colleagues at Geneva. And this is an open access data set available on Open Organelle. And you can see on the right-hand side, this is the exocrine tissue with these very large granules. And on the left side, you see the beta cell that is then surrounded by the other islet cells. And you see here, for example, an alpha cell with these very dark granules that don't have a halo. And we see now when we zoom out a little bit of this data set, is that it's really huge. It's a high resolution data set. We can also see these empty spaces within the islet are blood vessels, and we can see erythrocytes in some cases here. But we really have a fully preserved tissue context. And this, I think, makes these data very valuable. And we were actually screening these data sets originally to look into the interaction of beta cell cilia with cells that don't belong to the islet directly. So interaction with blood vessels, and also interaction with the surrounding exocrine tissue. So we zoom in now again to just see some of the structural features. And just to give you this overview again, we have this islet mass in the center surrounded by the exocrine tissue. We see blood vessels, and we see the different islet cells. And we screened these data sets to look into interaction of cilia with the exocrine, and this is what you can see here. And you see also here that the cilium is really squeezed in a narrow space between the exocrine tissue and its own cell. And we also interestingly found some cilia interacting with blood vessels, and this has not been shown before actually, because cilia are usually facing the avascular pole, so they are not involved in measuring blood glucose, but we found some cases where this happens. Now, this was not really done quantitatively yet. We just had some snapshots here and used some examples now from these data sets. And we turned actually to expansion microscopy in collaboration with Nikolai Kleiner and Gaia Picino at Human Technopole to get more quantitative data on these cilia interactions within the pancreas tissue. And Nikolai established here expansion of pancreas tissue slices. Um, and expansion is not the focus of this talk, but it's a nice tool to really get more quantitative data that can accompany then your structural data. And we were able then to, for example, quantify the percentage of ciliated beta cells. We could look into the length, in the cilia length of beta cells and non-beta cells. And we also could get an idea on how beta cell cilia interact with neighboring cells. And they were mostly interacting with other beta cells, also with non-beta cells, a little bit with capillaries, and then with the outside of the islet. And then some interactions we could not really identify from the expansion. And then coming back to this FIP data set, what was then exciting for me is, I have been used for years now looking into beta cells only, and I know how they look like, I know the structural features. But when it then comes to other tissues, like even blood vessels or the exocrine, this is getting a bit tricky. But when we look closely into these FIP data, and I talked a little bit about the innervation of islets before, we also can see axons penetrating the islets. And you will see these soon appearing in this movie here. So we have, for example, some cells here going along these beta cells, and these are actually neurons with synaptic vesicles, with microtubule cables running through. And what was so surprising to see here, and also quite exciting, was when we see here a basal body of the cilium now in the center, that in some cases cilia of these beta and also alpha cells were contacting these pancreatic neurons. And these were like transient contacts, but they were definitely touching. And also in some cases, we could see structural features of synapses at the site of the neurons here. And this reminded us a little bit of this paper, and this has been described only in the brain before, that cilia of neurons can contact axons and form these axociliary synapses that then drive changes in the nuclear signaling and chromatin accessibility. And the cilium is really a distinct signaling compartment. So it also, the signaling that is running through the cilium differs from the plasma membrane. So it's quite interesting to think of the cilium also as this special compartment receiving now signals from the nervous system, especially in the pancreas. And we found now some more cases in this data set. And maybe you can see this here when we now run through, we have a cilium here at the bottom that is then contacting this neuron here, and we have a presynaptic terminal here. And we can see this in the still image that we have this blob here with mitochondria and dense core vesicles and also synaptic vesicles, and then a cilium here that is in contact with those, with this presynapse. And just to have a better look at this, this is a 3D rendering of the region that I showed you in the previous slide. So we see the cilium of the beta cell in purple, and it's in contact with this transparent axon, with this presynaptic structures here with mitochondria and with synaptic vesicles. We have then looked also by fluorescence microscopy and tried to identify the neurotransmitters that are involved in this. And what you can see here are confocal images of cryosections of islets, of pancreas tissue actually. And in white, you see beta-3 tubulin, which is a staining for the nerves. ARL13B in cyan is a specific marker for primary cilia. Insulin you see in green, and synapsin 1 is a marker for presynaptic vesicles. And you see here in panel 1 and 2 that we find cases here where cilia are really in contact with nerves. And at most of these contact sites, we see synapsin 1, which is an indicator for an active presynapse. We also could define the neurotransmitters that are involved in the signaling. So we could see that acetylcholine is present at these sites. What we don't know yet, if these are really functional signaling compartments, And coming a bit to one of the open questions, this is what Patricia Santos is actually working on, finding out if these CR neuron contacts are functional and if they are, for example, involved in the cephalic phase of insulin secretion. So insulin secretion is also regulated by neurotransmitters, and actually acetylcholine is responsible for this first phase of insulin secretion that appears when there is no increase in blood glucose levels, but when we perceive the smell or the taste of food, for example. And then one of her questions is also what makes the signaling pathway special compared to signaling through the plasma membrane. So really, we found with the help of volume-EM like a new structure in the pancreas, and we are now looking into defining this better. And Solange, our co-chair in the lab, she is actually looking now into these large volume-EM data sets and trying to map all the connections in the islets, not only focusing on the cilium, but looking into all of the connections between cells and their signaling compartments, and also ask the questions if these are altered in diabetes. I made one video yesterday because what one of my messages is in general to spend a lot of time with the raw data. So we all know that segmentation tools are really important and we cannot live without them anymore, but we also need to spend time with the raw data. And what is really nice these days is that a lot of these data are being made open access available. And one example is Open Organelle. Empire is another example. On Open Organelle, you can find volume fib-SEM data actually of different cells and tissues. And I'm just running this video now just to give you an idea, because these are now actually, there have been made a lot of effort now to really make those accessible. And you can view these data sets in Euroglancer directly in the browser. And this is what we are looking into here, right? That we can now, for example, browse these pancreas data sets, but there are now different tissues available, kidney, liver, for example. And if we are going back now, we can also copy a link that we can then open in Big Data Viewer in Fiji. And this is actually what I was doing all the time when I was screening this large data set. So when we now open, so we copied now the link, we open Fiji. We go to Big Data Viewer and we paste the link here, and we can detect this data set. And this might take a while, but we can then look into the raw EM data. In some cases, we also have access to already some segmentation data here. But we can now open this data set in Big Data Viewer. And this is like in a ZAR file, so it's a chunked format. So we have this whole data set available for us, but we don't load all of it into the RAM. And we can now browse through the data in Big Data Viewer. And if we now find, for example, some interesting regions that we want to look into further and also locally on our computer, we can just extract this volume to Fiji and then save it locally, work on segmentation. So this gives us really this opportunity to look into data and not necessarily have to acquire data sets on our own for all of our questions, because a lot of these data sets are already there actually. So, and I'm coming now already to this take-home messages. So I hope I could convince you and also show that it's important to spend time with your data. That is also, we need to make use of these open access data sets because they are there, and since it's open access, we are allowed to use them. And if we cite them properly, everything is fine. Also looking into these structures, I mentioned that I have been looking into beta cells for more than 10 years. I haven't been looking into neurons, but I need to learn a lot still, and there are a lot of classical papers from the 60s and 70s and also books on ultrastructure that are really helpful in this. So you can really prepare to see strange things in these complex data sets and find new connections, new interactions between cells that we could not see before only in 2D data. Coming now to the end, I just want to show one of these animations again that again highlights these complex interactions. So we have a blood vessel here in red, and we see some axons here squeezing in actually between blood vessels and beta cells. We see some presynaptic structures and an axon then in contact with one of the beta cells here. So I'm at the end of my talk, and I want to acknowledge all the support from my lab and from the collaborators, especially at Genelia and Yale, and also at the Human Technopole in Italy. Also locally, people in Dresden have been supportive through the years. I want to thank the funders of my research. I want to thank you for your attention.